Hello, everyone. We have Dr. Marijin Lijffijt here with us today, and he is a translational medicine expert for the central nervous system. He has a PhD in neuropsychopharmacology, and he's done research across a broad range of domains, and he's currently working as a biotech and pharma consultant. First off, I'd love to hear your story, Ryan.
Yeah, absolutely. Thanks again, Robert, for inviting me. I really appreciate it. It's just a lot of fun to talk about biomarkers and pharma and biotech. Yeah, a little bit about my background. I'm a PhD in neuropsychopharmacology. I got that in 2004 in the Netherlands. And neuropsychopharmacology basically is about studying the relationship between brain and behavior, which is very broad. The way that I've always done it is, first of all, in human mental illnesses. And secondly, I have used several tools for that, including EEG, where you can measure brain activity from neural cells, from brain cells. But I've also used medication to change brain states in some people and then measure EEG as well as their behavior, including all cognitive tasks that can capture attention, response inhibition, which are thought to relate to very basic mechanisms of human behavior. I've always been in the academic world until a year ago. I wanted to be closer to really the development of interventions. I had a lot of fun in academia. I worked with some brilliant people and with a lot of different illnesses in people. But I had the impression that I wasn't contributing enough and contributing fast enough basically to developments. Therefore, I was looking at the industry, and I was fortunate enough to be hired by a company in the pharma field, which was Sama Therapeutics, and there was a great experience. Unfortunately, they had a reorganization 4 months ago, whereby 40% of the staff was laid off. Luckily, I was picked up by Sama Therapeutics, and both at Sama Therapeutics and Sama Therapeutics, I basically still do the same thing that I did before. I work with EEG and other tools whereby we look at biomarkers of different biological trajectories and associations with an illness. So what I mean by that is that an illness, for example, depression, can have different biological causes. And with EEG, ECG, wearables from the cell phone, fMRI, TMS, we try to extract proxies or biomarkers or characteristics that are related to the different biological mechanisms that basically underlie the different illnesses. So when you are able to do that, you can distinguish between different biotypes. And I think if you look at mental illness, it's very good actually also to look back at some other fields, including oncology, where there has been a huge success in defining biomarkers, using biomarkers to define different biotypes, and by extension, using those biotypes to start guiding treatment. And that is basically what I've always been interested in and what I've been doing in my academic career, as well as Sage and Sama, to see, okay, how can we extract those biomarkers? How can we measure those biomarkers? First of all, to show that the medication targets the right molecules, as well as that it targets the right mechanisms underlying an illness. And are those mechanisms specific to one biotype of an illness or perhaps cross illnesses? So it's a huge collection of information basically, but that's basically my trip across the years and where I am right
Wow, it seems like it's been such an interesting journey so far, and really exciting it to see things, both from the academic side as well as from the biotech startup side. Something that really stood out to me is, especially nowadays, we're collecting so much data about ourselves through these things like wearables, EEGs, and everything, and then the machine learning and AI is developing at such a rapid pace. It seems like it's just the perfect time to be mining for these biomarkers and just learning more about the body, learning more about the brain, and trying to develop the best therapeutics that we can. And after hearing all of the interesting points of your story, I've got lots of follow up questions. And the first one that I wanted to start with is, how did you transition out of academia and into the biotech startup world? Was it something that kind of just happened naturally, or was it something that you had started planning a little bit in advance? And what are some of the differences that really stood out to you when you first made that transition?
Wow, that is such a good question. One of the most difficult questions, I think, for me to answer. I was really starting to think about transitioning during COVID, where I was getting a little bit older. I was approaching by that time 48, and I was thinking, okay, what am I feeling that I am contributing everything that I can contribute to making life better, basically, for people. Now, even though I'm a PhD, but I've never treated patients as a therapist or something else. So it's always been in the research field, helping people through care. Direct care was not an option for me. And all the work that I was doing in the academic world, it was great. I mean, there is just so much knowledge that's being gathered, but it's very difficult to make basically one punch and go for the development of a novel treatment. In particular, with my background, neuropsychopharmacology, the intervention is very often medication-wise. So those two things, not seeing any patients, and at the same time looking to make to develop an intervention or trying to help develop an intervention, I really wanted to be more on the help intervention side, and I was not getting that enough, basically. And when I say not getting it enough, I think I was not searching for it perhaps enough within my position that I had in academia. I was doing clinical trials, so I was a PI, principal investigator on clinical trials, and I really liked that. I love the interaction with people, and I love the operational side and the paperwork side, and so I really like that. I also like designing studies. I've done that for a long time, designing studies, enriching that with biomarkers. So with that interest, I really started to develop the idea. Okay, I really want to make the jump to the industry, and that really I accelerated that about a year and a half ago, and then it took me about, well, a good 10 months, I think, before I landed a position. So it took me a long time, even though I thought, well, with my academic background, a lot that I do looked on the face of it quite similar to some of the descriptions that I found, listed in job offers and in job descriptions. But even with the background that I have, man, it was difficult eventually. I came into the industry, and one of the most difficult aspects I found was getting to familiarize myself with all the nomenclature and all the abbreviations that they have, CDT, RDLT, whatever you have. That alone, trying to navigate that aspect, and then the politics related to working with different aspects of an organization. I was basically used to keep everything in my own hand. and say, okay, this is what I want to do. I start a process, and I couldn't do that anymore. It was at least 1 or 2 times that my direct supervisor said, well, what you try to do here is start a process, but we cannot just start a process. You need to take a step back and approach that differently. So, okay, so I cannot start a process or procedure. So how do I then phrase it, and how does it fit really into the whole politics of an organization? And I don't mind politics. I was used to academic politics as well. I mean, I think you have a certain level of politics in any organization, and it can actually be a lot of fun to try to navigate that. But man, it was more difficult than I initially envisioned, and it was very, very humbling, basically, to be placed in that position. And I was just fortunate, both in my academic career, as well as when I started in the industry. I had great mentors, people who were brilliant, very enthusiastic. I love people who are direct, even though I like directness. I like people to tell me, okay, this is how it is. At the same time, there's always that little voice in my head that says, oh man, that's not good what you do there. And it reflects bad on me. So at the same time, I then have to reframe that and say, okay, I actually want that direct critique because I want to get better. I want to be better at what I'm doing. Man, it was definitely a very humbling experience, and it still is. It still is. I still learn so many new things because there are a lot of things the same. But for example, we have standard operating procedures. There are organizational policies. Some of the policies are the same. Let's say good clinical practice, HIPAA, FDA regulations. Those kind of things are all the same, and they're still being trained in both organizations. But I think it is mostly that I how do you say that? I basically have to take a step back in seeing myself in a leading position, because all of a sudden I was more in a learning position again.
Yeah, that makes a lot of sense, and totally agree with the importance of mentorship all throughout the career, from the early stages up through middle and even final stages of career. Mentorship is important all the way through. And then just switching between positions or even switching between industries, I totally understand how it could feel like you're drinking from a fire hose and just learning so many new things at once and trying to grab onto all the important information. So let's dive a little bit more into your work with Sama Therapeutics. So you mentioned that they're working on deriving biomarkers from things like EEG data and wearables. I was wondering if you could just share a little bit more detail about the technical side of that to the extent that you're allowed to. And I guess maybe a few questions to start us off could be, are there any specific either diseases or treatments that you're looking at biomarkers for? And then also, just from a high level, what is the process that you need to go about when deriving these?
Yes. Let's see where I can start the best. So Sama Therapeutics, I was onboarded as a consultant in September last year. The great thing is it's a startup. It's a very small organization. They work with a lot of consultants, but people are very enthusiastic, and the CEO is very energetic, and that rubs off. So whenever he's enthusiastic, it's just great to see him, and it's great to hear his vision about the company. And one of the aspects that we can begin to do. So like I said, I worked for a very long time with biomarkers, and for a very long time, I was trying to define biotypes. And I've done it with EEG by looking at the difference between people with an illness and without an illness, so-called control group, or difference between two types of different illnesses, let's say anxiety and depression. And then you can also look at at the flip side, so you have the biotypes of an illness itself, but then you have also biotypes related to a treatment response, because not everyone who has an illness responds the same to just any treatments. Now, I've worked a lot in the last 10 years in my academic career, but also at Sage and now at Sama again, with modulators that act on cortical inhibition and excitation. You must think about NMDA receptor modulators, antagonists like ketamine. And at Sage, we work with GABA modulators, GABA receptor positive allosteric modulators, to have been approved for postpartum depression in the last couple of years. And in Sama, again, I'm working more, one aspect at least, on a ketamine side. The reason why ketamine has gained traction over the last 10, 15 years is because regular medications like SSRIs that a lot of people with depression and anxiety get are just not cutting it. There's only a treatment response of about 30 to 40% of the people. And treatment response means that people feel about 50% better. I mean, they don't feel like they are totally healed and totally better. You know, first of all, the medications don't heal anyway. If you really go deeper, then you have remission, and that's even fewer people that meet that. So what happens in practice is that there is a trial and error aspect. People come into a clinic either with a history of depression or with new onset depression or anxiety or PTSD or whatever you have. And there is a list of medications, and that's partly determined by consensus from experts in the field, as well as consensus with the insurance company that says, okay, we are going to try an SSRI. And not all SSRIs it stands for serotonin, selective serotonin reuptake inhibitor. But not all SSRIs are the same molecule. So they tend to have slightly different effects on different types of molecules, even though the main focus is serotonin. Now, the trial and error, and I went through it myself about 18 or 20 years ago. Started off with a medication, and I was sick, and it didn't help at all. I was just sick. I was literally green in the face, and that happens to a lot of people. So one medication is being given, it doesn't work. You step to another SSRI that doesn't work, or work a little bit. If it works a little bit, you increase the dose, or you add a different types of SSRI to it, or a different medication. So eventually there's a lot of trial and error. A lot of people experience some improvement if they do after some of the acute side effects disappear. And it can take about 4 to 8 weeks before people really feel starting a little bit better. It would be great if we actually had some biomarkers that can determine beforehand what treatment people could have, and what the chances are that the treatment can actually work. So what we do at Sama is actually several things. We are trying to develop product that can that uses artificial intelligence and machine learning to analyze features in EEG data that can be collected when people come into a clinic, but also ECG data, so heart rate data. And we can also think about some other types of data that we will include, whereby the features are being extracted. And you need to think about 200 features from just EEG that is collected for a certain period of time, and ECG. The machine learning and artificial intelligence algorithms start trying to cluster the information in such a way that you can build a biomarker model. Now, first of all, you do it on a global, on a broader population. But eventually, if you have more than enough data in there, you can start looking at individual people and start predicting, okay, this is a certain biotype that we found before, and we show that that biotype reacts relatively well for this particular SSRI or for ketamine. So, or there's another side, this this particular profile shows that people respond relatively badly to this particular medication, like another SSRI. And that's basically how Sama wants to use biomarkers and artificial intelligence to start guiding treatment. Now, beside that aspect, there's also an arm of Sama that is really looking more at the clinical trials. How can we use, there are a lot of data that is collected in clinical trials, primarily clinical data like symptoms, but in early studies, there's also a lot of collection of EEG data, just to show that a medication is actually getting into the brain and that medication is engaged with a particular molecule. For example, ketamine, an NMDA receptor antagonist, can acutely increase a certain portion of the EEG, the gamma band. And that is taken as a sign that it engages with the NMDA receptor, either through the NMDA receptor or another type of glutamate receptor. So that's basically a first step that a lot of companies can use EEG or other electrophysiological or wearables. Then down the line, there are other biomarkers that can say, okay, the first molecule has been engaged, but does it also engage and change mechanisms that we think are related to depression? For example, reward mechanisms or emotional regulation mechanisms like the amygdala with the frontal lobe. So that's basically how you can start using what the industry often does with those biomarkers. And what Sama wants to do is take all those biomarkers and again extract the 200, 300, 400 different features and then start clustering what biomarkers best predict differences between medications, between doses, as well as between a test medication compared to a placebo. And it's not always so obvious as you think, because a lot of the biomarker development is based on hypothesis. The hypothesis is that, like I said before, the gamma band in the resting state EEG will change, and that is a sign of NMDA receptor engagement. And then we expect that an emotional control mechanism will change. But what if the medication actually works, but not on those mechanisms? Then you basically miss a whole thing, even though the medication may still well work. So what Sama is doing, also in the clinic, but also for clinical trials, is we take an agnostic approach. It means that it is hypothesis-free. We collect information and then say, okay, we just let the algorithms sort the data and work with the data instead of us really trying to look with a particular eye on the data. And we can use resting state EEG for that. We can use particular paradigms for that, which is a little bit more in the field again of hypothesis testing, but especially the resting state EEG can be a very powerful tool. I hope that answers your question a little bit. I quite long-winded, but
Yeah, that absolutely delves into what I was hoping to understand further by my question, and that's a really exciting mission that Sama is working on. And I've heard of similar. I've heard of companies trying to do similar things via genetics, and something that really stood out to me when you were talking about Sama is that it's a lot more. It's something that can be done more times than one as the person is changing and as the medication is working on them. It's something that it's not static, whereas like certain genetic testing for these similar things is static. And so I think that's awesome to be tracked over time and super interesting, super exciting, and wishing all the best with the development. And then another thing that stood out to me there was how you mentioned that you're collecting lots of data, feeding it to the machine learning and AI algorithms, and kind of letting that sort out the data on its own and see if it derives any insights that maybe the scientists working on it may not have thought of that hypothesis. These algorithms are so powerful these days that it allows us to look at data in thousands or millions of ways that we may never have even considered looking at it before. And so that's that's awesome that you guys are working on that.
Yeah, that's it's a very exciting time to live in. There's so much data out there that has been collected throughout the years since basically the conception of internet. Data has become more and more available. It's just amazing how the information can be used now that AI and machine learning is really being developed a lot further. And I mean, artificial intelligence and machine learning are not necessarily new, because people have been working on it since the last, for the last 20 years with neural models and those kind of things. The way it is progressing right now, and how it can handle those huge data sets, and to look into data in a way that we haven't been able to do before. I mean, it's amazing that some of the dreams that we had can actually be not necessarily come true, because I think it's a little bit too early, or much too early to say that, but that the dreams can actually be lived, and that's just amazing.
Yeah, it really is amazing. In some ways, science fiction is starting to become reality. Zooming out a little bit more, looking towards the future, do you have any predictions for the future of neurotechnology, subset of neurotechnology, where we're pairing it with drug development or pharmacology or recognizing disease states and looking at biomarkers, and then even just in a broader sense for the field at large?
Wow, that's a very difficult question. I'm not sure where I read it, but I believe most predictions about the future don't come true. It's what I think, if we look at oncology and arteriovascular disease and development for medications they're a lot further than mental health, and I also think than a lot of areas in neurology. In that aspect, we are really still, and it's horrible to say, but even after 100 years, we're still so at the beginning of understanding mental illness and what it means, both at a society level, individual level, and medication development or treatment development. I shouldn't always say medication, but treatment development level. But if you look at oncology, the way that people have used biomarkers there, and also artificial intelligence now there, they have done a great job to more and more zoom in on the different genotypes and biotypes, and actually develop treatments for those specific genotypes and biotypes. And that is basically a vision that a lot of people in mental health, researchers and treatment providers have, to you get someone into your clinic, and it's always easy to ascertain what people have for type of disease. I mean, the only thing you have to do is basically ask. And perhaps not every disease, but for example, depression, you ask, do you feel depressed? I mean, you don't have to necessarily have a biological test to know that people are depressed or are suicidal or have PTSD or have schizophrenia or OCD or whatever else you want to say, or an addiction. There is enough known that all those disorders are not homogeneous disorders. There's not, unlike some rare diseases, there's not just one genetic defect or genetic change that predisposes people to a certain mental illness. So the illnesses are very difficult, and they are in part difficult because when you look at a full group of people, you have all these different potential biological mechanisms that predispose to the same symptoms or the same symptom characteristics. It's the same in oncology, where you say, well, someone has cancer. Yeah, someone has cancer, but not one cancer is not the other cancer. But in mental health, we still do it. I mean, we say someone has depression, but one depression is not the other depression. And sometimes you can just not only describe it by symptoms. If you look at only symptoms, it seems that the symptoms are not really capable of just driving and guiding treatment. So you need to look at a different level. And hopefully biomarkers and biology, especially biomarkers that can be collected relatively easy, can be collected when people are just doing their everyday things. I mean, you don't necessarily have to think about EEG, but you can think about EKG, number of steps that people are setting, the way that people talk, the way that people's muscles are changing in their face when they communicate with people. Those are all things that nowadays can be collected. And some people, they are collected for some people because they sign up for those kind of things. And people have an Apple Watch, or people have something else, and all that data is potentially very rich to start teasing apart biotypes. And it is not likely to be so. And that comes back to your question: what brings the future? Just like genetics, it is unlikely that one biotype will solve the biotype issue. So one biomarker per biotype is very unlikely. We would probably already have been there if that was the case. So it's probably a whole construct of different biomarkers that starts to predict a certain outcome. And I think in the years to come, the models of predicting the outcome will become better and better. People's individual data will be more embedded in data collected from a bigger population, where more and more people sign up to share their data anonymously, so that models can be optimized.
Yeah, I totally agree. Like in some ways, we really are kind of at the very early stages of understanding mental health, but hopefully, as you mentioned, there's a bright future ahead as we start to collect more of this data and just understand more about ourselves.
Yeah, absolutely. It's just amazing how that some of the brightest people are working in the field of understanding mental health, treating people, breaking down stigma. I mean, there are so many patients or people living with an illness who are saying, okay, this is part of me, and I'm actively searching for a new intervention, a new treatment, and people are working very hard. And any new technique can help to basically make that step in the right direction. So I think the most important thing is that, and I go back again to depression, which you also have with anxiety and some other disorders. The purpose is not for people to be all happy all the time, because people are not happy all the time. But the purpose of a treatment is that people can live their lives the way they want to, with their ups and downs, but ups and downs don't go either too deep or too high, like you have with mania, and too deep with depression, or and people are able to cope with it, and their illness is not guiding their life anymore. And I think that is really something that treatments are trying to achieve. And if we can get to the right treatment quicker by having the right biomarkers and defining the right biotypes, and working on developing treatments specific for a biotype, for example, by including a particular biotype in clinical trials, so-called stratification, that is basically a way forward. And I think that will be more and more what will be done in the next 10 years, I think.
Absolutely, and it sounds like a great path forward, and wishing best of luck to you and the rest of the team at Sama on working on that. Once again, we really appreciate having you on, and I feel like we've learned a ton from you today. And before signing off, I wanted to ask, can our listeners contact you after the episode for further discussion? And if so, how could they reach out to you? And what kind of things should they reach out to you about?
Yes, no, they can absolutely do that. They can reach out to me through LinkedIn. I think what you can do is just put my LinkedIn account there, and they can message me and connect with me. That is the easiest way to do. They can actually reach out for two different things. I love just to chat about anything, so I'm always open for a virtual coffee. Also, and I'm not sure if that's appropriate, but we are looking for angel investors for Sama, meaning people who want to invest $5,000 to $10,000 for a company that has products that's already selling products to clients, and that is about to go into the seeding rounds in the coming months. So they can also reach out about that, and they can people can get a pitch deck, and people can connect either with me or the CEO of Sama.
Well, great. So everyone, if you want to have a virtual coffee with Ryan, or if you're an angel investor looking to expand your portfolio in the biotech, mental health, biomarker space, feel free to reach out to him or the CEO over at Sama. And thanks again for coming on. We really appreciate it, and it's been a super interesting conversation. And wishing you all the best.
Hey, thank you, Robert.
Hey, thank you very much. Goodbye.
Bye.